Okay. So the sight and smell of the food releases gastric fluids in the stomach, which include mucus, hydrochloric acid, and pepsinogens. Incisors cut and molars crush the food, beginning the first stages of physical digestion. Saliva is released from the salivary glands, and the enzyme amylase begins to break down the starches in the food into simpler sugars. The food is chewed and formed into a bolus, where it is swallowed into the esophagus, and the peristaltic movement of the muscles in the esophagus move the bolus to the lower esophageal sphincter, or LES. The bolus enters the stomach through the LES. The bolus of the food contains sugars, fats, and proteins. The walls of the stomach are distended by the presence of food, causing the hormone gastrin to be produced. Gastrin simulates the hydrochloric acid release in the stomach. The presence of hydrochloric acid in the stomach converts the zymogen pepsinogen into its active form, pepsin, which begins to break down the amino acid chains in the proteins of food into polypeptides. The proteins of the stomach cells are protected by a layer of mucus and not affected by pepsin. Any vitamins that may have been present in the food can then be absorbed by the stomach. The chyme of the partially digested food from the stomach enters the small intestine, specifically the first part of it known as the duodenum, via the pyloric sphincter. At this point, the pH of the chyme is around 1 to 2. Prosecretin is converted into secretin when the acidity is present in the duodenum, and the secretin is absorbed into the bloodstream, where it is carried to the pancreas. The pancreas releases bicarbonate ions to lower the pH to a more basic level, around a pH of 9. This basic pH is inactive pepsin. Inactivated in the pancreatic secretions is the trypsinogen. When trypsinogen reaches the duodenum, it is activated by enterokinase into trypsin. The trypsin breaks down partially digested pepsin proteins from long-chain polypeptides into smaller peptides. Erepsins that are also released from the pancreas further this digestive process, separating the short-chain peptides into individual amino acids more suitable for absorption. The digestive process of starches that started in the mouth is continued here, as well as the pancreas releases amylase, breaking down the polysaccharides into disaccharides. This meal is partially fatty, so enterogastrin is released by the small intestine to slow the peristaltic movements of the esophagus so that more time is allowed for fat digestion. Fats are now present in the small intestine, causing cholecystokinin to be released and absorbed into the bloodstream, which is carried to the gallbladder. The gallbladder then releases bile salts, which acts as a stage of physical digestion for fats and preparing them for chemical digestion, where it emulsifies the larger fat globules into smaller ones. Once the larger globules are broken down and the surface area is increased due to this emulsification, pancreatic lipase and phospholipase released by the pancreas can then work. The food reaches the large intestine, where it has completely gone through the stages of chemical digestion and the majority of its nutrients have already been absorbed. As it moves to the colon, it is stored there long enough for the reabsorption of water. Some salts, vitamins, and minerals are also absorbed here. Cellulose present in the cookie arrive at the colon undigested. Cellulose provides a mass to waste stored in the colon, causing a bowel movement to be prompted through the receptors in the walls of the large intestine, detecting the buildup of waste. The wastes and toxins are then excreted out of the body via a bowel movement, ending the digestive process of the food.